Why don't we get started? No, Pete, don't take that. Come on. No, okay, if you're going to be more comfortable, you should take that. I'm just frankly, it's so novel to me that I figured it's more.
Fix your collar.
All right, good afternoon. So it's my pleasure to introduce Pete Mulligan to give his defense today. I was kind of thinking a little bit about what my experience has been with Pete, and frankly, it's really hard for me to imagine in my head what having a lab is like without having Pete in the lab, because he was one of my first students, and he came from MIT, very talented guy. Had an NSF graduate fellowship, was ready and raring to go. And I think that when you're starting up a lab, you owe a great deal to your first students, because they really are, they're taking a chance on you in a lot of ways, and him coming from that background and having all that talent is it's just a real it's a great pleasure and a real benefit to a young faculty to have somebody come into the lab like that. And so he jumped in and he took on a very, very challenging problem. And I think that what's kind of tricky about what you're going to see him talk about today is that since he took on a really challenging problem that was new in our lab, he had to grapple with some of the hardest issues in getting a model that actually had the right features in it, okay? And he had to scratch his head, and I had to scratch my head, and we had to come up with this model from scratch. And so I hope that you appreciate just the inherent challenges that you face when you have to develop something like that from the beginning. So what happened was he did some very, very nice work. It took a long time, and we were ready to publish on it. For the first time, we were really excited, and then I got a call from Rob Phillips that he had some experiments that he measured, and about 2 months later, we had a whole new model for his experiments that he was publishing on. So it's like he had all this time of this really hard, challenging problem that did bear fruit, and we're really excited about those results. And then there was a second project that he's also going to talk about that was so much faster, and he could leverage all of his development on that type of problem. So I'm really excited to hear what you have to say today, and let me just leave it at that. So, Pete.
All right, well, thank you everyone for coming. Thank you, Andy, for the nice introduction. As Andy mentioned, I'm going to be talking to you about two of the projects from my work here at Stanford. The first is going to be on the condensation of these dense regions of DNA called heterochromatin. And the second is going to be looking at looping kinetics of DNA. And so every person, every organism has a unique genetic code which is captured in the base pairs of your DNA. But in order for this information to then control the different functions of the cell, it has to be accessed, transcribed into RNA, and then translated into proteins. But this whole process is happening within this crowded cellular environment. We can see here there's lots of molecules going to be interacting, and the question I'm going to be really addressing is how the physical properties of these different molecules affects the access and controls access to the DNA. And if you can imagine the DNA, inside one of your cells, the DNA is packed. You have 2 meters long DNA packed into this very tiny volume. And so there's several levels of compaction going on, from DNA wrapping around proteins into these fiber structures and into higher order structures. And there's still a lot of discussion and debate about what these structures are inside the cell. Today I'm going to be talking to you about regulating access to DNA across 2 different length scales. The first is for these short segments of DNA, looking at how DNA looping can help regulate transcription. And then the second is talking about these very dense packings of DNA at these sort of larger chromatin scale. And the way I'm going to be approaching this problem is using physically inspired models to try to capture the essential features of these really complex biological processes. So now is really an exciting time to be working in this area, where we have single molecule experiments like these that can measure the properties of these different molecules. So in this one, you can attach a DNA to a bead, and you can stretch it, you can twist it, and you can measure the response and get a sense of what these molecular properties are. In addition, we can get a picture now of the in vivo conditions. And in this example here, you can look at the organization of DNA by attaching different regions to each other, and then mapping which regions of the chromosome are contacted with which other regions, giving us a picture of some of the in vivo organization of DNA. Now ultimately, our goal is to take our physical models and be able to interpret and provide new testable predictions to the experiments, as well as to get a sense and what are the key features inside the cell that allow these properties to happen. Now ultimately, the goal of such fundamental research is for advances in medicine. And here's an example that's particularly relevant to the work I'm talking about. In this picture here, a tumor suppressor gene is active when it's located within this open conformation of chromatin. And this open conformation is has these acetylation marks in it. Now, the loss of acetylation marks can lead to the condensation of the chromatin region, and ultimately repressing this gene and leading to cancer growth. We can also use our understanding of DNA organization to develop new tools for medicine. So in this example, someone with a genetic disease has a mutation in a key gene, and one of the ways to treat this is to deliver the corrected gene to the patient, a process called gene therapy, in which we need to then pack the DNA into a very small containment vessel to deliver it to each of the cells. And often that containment vessel might be a virus. And so understanding the properties of DNA and how it's packaged can help us design better delivery vessels for gene therapy. Now today, as I mentioned, I'm going to be talking to you about two problems. First, I'm going to look at how proteins can mediate the looping process for DNA. And then in the second half, I'm going to talk about these very dense areas of packing called heterochromatin. So first, I'm going to give you some motivation for why we're interested in DNA looping kinetics, talk to you about our elastic model, and then talk about some comparisons to experiments, as well as what the key effect of protein flexibility is in these systems. So first, looping is actually a really common process used throughout the cell. And it's used in a number of different areas. Looping can help regulate the access to the genes in this region. Supercoiled loops can form, and this helps facilitate packing within organisms like E. coli. And looping can also facilitate the spread of silencing marks or chemical marks along the DNA. And this is something I'll talk to you a lot about in the second half of the talk. But right now, I want to focus on a very canonical example, that of lac repressor protein. That's in green here. And the lac repressor binds two sites on the DNA, and this helps stabilize the formation of a DNA loop. Now, this process is very important for the regulation of the genes in this region by blocking the initiation of transcription and the RNA polymerase. And so, the question I'm going to be addressing is what role do the physical properties, like the elastic properties of both the protein and the DNA loop play in how long it takes for these loops to form, and then how stable and how long these loops last. Now, as Andy mentioned at the beginning, this work was also motivated by a series of experiments by our collaborators at Caltech. And what they did was they attached DNA to a cover slip, and then they tethered the bead as well. And they could watch the motion of the bead. And they added these binding sites for lac repressor, so that when you add lac repressor, sometimes it's going to bind here and cause a loop to form. And because of the loop formation, the bead motion is going to be restricted. And so they could watch the motion of the bead and then see and identify whether it was in the unlooped state or the loop state based on the motion of the bead. And from this, they could measure how long it lasted in each of these states and calculate the lifetimes of those states. They could also calculate something called the J factor. And the J factor is essentially a measure of the probability of forming a loop. And it is really accounting for the other part of this process here. So you have the lac repressor binding to the other site B, but then the DNA also has to deform itself into a loop. And so the J factor is really just a measure of how much the DNA has deformed. Now I'm going to show you a plot of the J factor versus loop length that comes from our model. And I'll explain how we get it in this a little bit later, but this is to give you a sense of what the J factor looks like. At very short lengths, the looping probability is going to be low. And that's because the DNA is very stiff and hard to bend into that. At very long lengths, the J factor also decreases. And this is because you have a high entropic penalty for keeping two ends that are separated by far distance close together. So now I'm going to talk to you about our elastic model for this process of looping. And we're going to consider three main energy contributions. The first is going to be bending of the DNA. The second is twisting to orient the binding site to be able to bind to lac repressor. And then the third piece is the actual binding of the lac repressor protein. And within this piece, one of the key sort of novel features of our model is we're going to account for the flexibility of this protein. And I'll explain how we do that in a bit. But let's start with the bending energy. So DNA is a stiff molecule. And that means that for short lengths, for example, around 100 base pairs, the DNA would prefer to take more of a rod-like conformation. And this 100 base pairs is something that was measured in the experiments as well as common in the in vivo system. And so we can actually account for the bending energy by looking at how the tangent vector, u, changes as we move along the chain. And we can assign a penalty, a quadratic penalty, to the change in the tangent vector. And that's essentially accounting for the more the tangent vector changes, the more the DNA chain bends. And this model is called the worm-like chain model. And it has a parameter called the persistence length, which is essentially the resistance to bending. And it's also just basically the length at which DNA would prefer to look like a rod. And we'll see its effect in a second. So if we integrate over all the possible configurations of the end-to-end distance, we can get the free energy. I'm going to plot the free energy right here in green versus the end-to-end distance. And if you look, there's at very large extensions here, there's a high entropic penalty because the two ends, there's not a lot of configurations that they can take. There's a minimum, and now we can see the effect of the persistence length. The chain at 34 nanometers is very close to full extension at its relaxed state. And that's because the persistence length of this chain is larger than the chain length, so we prefer to be rod-like. Finally, if we bring the two ends close together, it's going to force a lot of bending penalties, as well as entropic penalties for holding the two ends close to one another. DNA also has a natural helicity to it, in which every 10.5 base pairs, it rotates a full 360 degrees around its axis. And so what this means is that for a given loop length, the DNA will complete some number of turns as it moves around the loop. And this means that the binding site for lac repressor is going to be located at some relaxed state here. Now, the binding site needs to be oriented to face the protein lac repressor. And so if after the natural location is away from the lac repressor here, the DNA would have to twist so that the binding can actually occur. And so we can account for this twist in the DNA by this angle theta, how much it has to twist. And we can just say that the energy of twisting is just a quadratic penalty in the amount of twisting that we have. And then we have LT, which is just the resistance to twisting. And so let's look at what the twist energy looks like. The twist energy here versus loop length. And you can see that when the binding sites are in the relaxed state anti-aligned from one another, the DNA would have to twist 180 degrees. And so the twist energy is at its maximum. But if at the relaxed state, it's already facing, there's no twist that needs to occur, so the twist energy would just be 0 in this case. The last piece is the binding energy. And we have a epsilon 0 binding interaction for the lac repressor to the binding site. And then we have an interaction distance delta. And this is where we are introducing the effect of protein flexibility. And so the larger delta is, the more flexible our protein will be. And I'll show you how this works in a second. Here is our binding energy. It's just a simple exponential well in which we have this quantity Ra. And Ra is to account for the penalty for being in the wrong orientation. And so we can see that in this diagram here. When the binding site is further away or more off alignment, the vector Ra here is going to be further, larger, and so the energy it feels will be less. And so if we look at what our binding energy looks like, we see that there's a well depth epsilon 0, and this is for the case where the two binding sites are aligned. And at this point, at this short end-to-end distance, the protein doesn't need to be flexible. It doesn't need to deform at all, because it can easily bind to the other site. However, if the end-to-end distance is further away, we're allowing that the binding can still occur if the protein has now deformed. And so the penalty for this deformation is basically the difference between the epsilon 0 that it would have felt and what the actual binding energy is. And that's capturing the effect of the flexibility of this protein, the penalty for deforming it. Now we can put all of these energies together on a free energy landscape, as shown here, in which we have the end-to-end distance versus the twist angle. And we start up here at the unlooped state, x, and we define a minimum energy path, black along here. And we can see what the different conformations look like along this pathway. So it's going to start, be very relaxed and stretched out, and then as the two ends come together, you see it bending, and then I also have color-coded the twist amount. So it only starts to twist as it approaches the transition state here, which is just the maximum height along this pathway. So we can look at a slice the energy along this pathway, and that's the black line here, the total free energy. And if we just want to focus in on this plot here, the total energy is just a combination of the bending, twisting, and binding energies. And you can see that both the twisting and the binding are only going to start happening closer to the transition state point. And the reason for this is that the twisting is only going to start to be felt once it starts to feel binding. It has no reason to twist if the binding site isn't close. And so we'll be referring to this free energy diagram throughout. So now to actually calculate our lifetimes, that is how long it stays in each state, we can basically solve a problem of 1D diffusion on this free energy landscape here. And so we have a number of conformations that it takes as it diffuses along this pathway. And the lifetime is really just going to be the survival time that it takes for leaving the loop state and crossing the transition state here. And similarly, we have the unlooped survival time to tell us the unlooped lifetime. Now these lifetimes we can solve for exactly from the 1D diffusion equation, but they're going to be roughly proportional to the barriers. So the larger the free energy barrier has to go over, the longer it will take to leave that state. We can also calculate our J factor. And the J factor is just accounting for the free energy difference only of the DNA molecule itself and not the binding piece. And so the J factor is just a free energy change in the DNA between the looped and the unlooped state. And here's what it looks like. The black one is the curve is the same one I showed you before for the J factor, where we plotted J factor versus loop length. And now you see the gray one, which accounts for twist oscillations. So when it's at a minimum here, it means that the twist, the binding site is anti-aligned. And so it's less favorable to bind if the binding site is aligned away from the lac compressor protein. So now I want to talk to you about some experiments that we compared our work to, as well as this key effect of protein flexibility that we introduced. So this is just to remind you of the experimental setup. We have a bead tethered by DNA, and we can watch the motion of the bead and look at the lifetimes of the different states. And we can take an average over all these, over many different trajectories to get the average lifetimes. And we can also vary the DNA in this region by changing the length, as well as the sequence. So here's just one sequence. plotted J factor versus loop length for 101 to 108 base pairs. And you can see that the J factor, in black here, goes up and down like we saw because of twist. Now the looped and non-looped lifetimes in red and blue here also follow a very similar trend to the J factor. And so if we just plot them versus the J factor, so we're going to plot the red and blue lines just versus the J factor quantity, and you can see that as you change the length from 101 to 102 back around to 108, it's going to come up and then go down because of the change in the twist angle that it has to go through. And notice that we have a rough linear relationship on this log-log plot. Now, it's one thing that's kind of surprising here is that we notice that the loop lifetime is dependent on the J factor as well. And what essentially the J factor is measuring is how much the DNA is deforming. And so in a traditional picture, maybe the DNA would deform all the way and then the binding event would occur. And that's sort of a traditional view of how this looping process works. But this dependence is telling us that there's some DNA deformation occurring in the looped lifetime state. And I'll explore that on this next slide. So here is a range of different sequences plotted and a range of different lengths plotted here. And just looking at the loop lifetime versus J factor. And we again see this linear relationship, which tells us that there's a power law scaling between the lifetime and the J factor. Now, I said that the J factor is capturing the DNA deformation. And so what it's really saying is that the process of going from this loop state to the transition state here is involving a release or a relaxation of the DNA deformation. And so the DNA is more highly bent in this state, it's much more loosely bent. And so the exponent is actually a measure of how much it's changing, how much the bending is changing within this process here. And if we compare this to the unlooped lifetime, we can see the unlooped lifetime is measuring this process of going from the unlooped state to the transition state. And it's going to have also a power law relationship with the J factor. And that comes from the fact that the ratios of the looped and unlooped lifetimes are basically a ratio of the probability of being in each state, and the J factor is capturing this looping probability. And what we see then is that these scaling laws of minus 0.48 and 0.35 are really telling us how much of the deformation is happening to go from the unlooped state to the transition state versus in the process of going from the transition state to the loop state. So it's telling us how much the DNA is deforming along the reaction trajectory. So we can look at how our model is able to capture this behavior. And so for simplicity, I put all the experimental data points in red. And then black line is a representative curve from our model. And we see it captures the essential features. You have the power law-like behavior that we see here. And then you see these oscillations, cutbacks, are the same ones we saw before where the twist was going up and down and adjusting the J factor. And this is for very reasonable values of these protein properties, which will be the same for all the experiments. The sequences, though, could have different properties of the DNA themselves. And so the persistence length that we find is well within the range of what's been measured. The twist persistence length is typically measured around closer to 100 nanometers. And so the deviation here in the twist, though, could be due to a very simple model for handling the twist, in which we only have one dimension, just that theta, where really we're dealing with a three-dimensional process of the lac compressor binding. And there are a number of other reasons, but the essential feature here is that we're able to replicate the experimental data for a number of different sequences and lengths. So now we can look at if our model can predict the correct behavior for a different set of experiments. And so another set of experiments that were done was to change one of the binding sites and switch in a weaker binding site, something that has weaker attraction. And in our model, this should really only affect one parameter, epsilon 0, the binding well depth. And we know what this energy change is from previous experiments, so we can just keep all the same parameters but shift down our epsilon 0. And if you look at the binding energy here, what you notice is that the binding energy is really only affecting the loop lifetime process. And so we'd expect only the loop lifetime should be affected by the change in the epsilon 0 term. And so here's the previous data I showed you in our previous model curve. Now in blue is the new data, and in black is the new model curve that just has a change in the epsilon 0 term. And we see, as we expected, that the loop lifetime is the only one that's affected. The on-loop lifetime stays the same. Another feature you'll notice is that the data, although it's a little noisy, it follows the same basic trends in both cases. That is the same rough scaling law. And I'm going to talk to you now about why that is and how important protein flexibility is in capturing this effect. So the essential feature of our protein flexibility is that when the protein is more and more flexible, it allows the binding energy to be felt at larger and larger separation, end-to-end distances. And because the binding energy starts to be felt at larger and larger end-to-end distances, the transition state here, and the heights of this barrier, are going to be shifted to larger and larger end-to-end distances. So this is the free energy versus the end-to-end distance. And we see that for the blue curve, which has the largest amount of protein flexibility, the largest value of delta, that the transition state is at the largest end-to-end distance. And so what this means is that the process of going from this loop state to the transition state is going to involve a larger distance at the separation, and that means there'll be less and less bending at this point. And so the DNA is relaxing more and more as the transition state is shifting. And this is, what this would suggest then is that the loop lifetime will be more dependent upon DNA deformation in the blue case than versus in the red case. And if we look here, that's exactly what we're capturing in this plot. So the blue curve, the highest amount of protein flexibility, has the greatest dependence of the loop lifetime on the J factor. And the J factor, remember, is basically a measure of the amount of DNA deformation. And so this dependence captured here is due to the flexibility shifting the transition state out to further and further end-to-end distances. But at the very short values, small values of delta, in which the protein isn't very flexible, here we start to see the picture that I discussed earlier, where all the DNA would deform all the way and then binding would occur. And so this picture would give you closer to a horizontal line where there would be no dependence of the loop lifetime on the J factor. If we look at the unloop lifetimes versus the J factor, we see again the opposite trend because of the relationship between the looped and unlooped lifetimes. And so what it's saying is that if more of the deformation change is captured in the loop lifetime process, well, then less of it will be captured in the unlooped lifetime process. And what you can tell from these plots here, and the gray curve is the model fit I showed you before, is that the scaling law that we captured here, we could not have captured that without incorporating the effect of protein flexibility. Right? If the protein was inflexible, then it would have predicted that it would have to come all the way before the binding energy was being felt. And so essentially what you're having is a trade-off between the amount that the DNA deforms and the amount that the protein will deform. And so this is the trade-off that's governing these relationships. So now I'm just going to conclude this section. And what we talked about was the different energies along the reaction pathway. I discussed how we were able to match the basic behavior from a range of different experimental results. How we outlined what the free energy landscape picture was for this process, and how the J factor is really measuring the dependence of the degree of deformation in the DNA. And finally, how protein flexibility is able to capture the change in this deformation from the unlooped lifetime to the loop lifetime process.